Hello, everyone. Welcome back to our question and answer period. And if someone would please let me know in the audience if you can hear and see us properly, that would be a real help. We'll just wait for that confirmation. Hello, everyone. Welcome back to our question and answer period. Yes. Okay. Ahmed is here. Yes. Good. Okay. So welcome back. I'm Steve Prescott. I'm here with Bill Bialek, and hopefully you were able to see his presentation live this morning, or had a chance to watch the recording of it. And he's back to answer your questions. We do have a few questions that were asked and didn't have a chance to get answered this morning. So I'm going to go to them first, but please, using the Ask a Question tab at the bottom, if you can please insert your questions there. At the same time, I think we have enough time that if you would like to ask question in person, still, insert your question using the tab. But then if you would like to, you can, indicate your willingness to come on screen and we can join, or you can use the comment bar to sort of, refine the question or respond. And hopefully we can do this seamlessly. So I will go to the questions and I'm just going to go in order that they're here. So from Apinder Bella, he asked, back to place cells, given that they remap, doesn't that scramble all the correlations?
Okay, so I guess the short answer is yes. If I'm understanding the motivation of the question, you're sort of asking, well, if you base an analysis, so that observation could be raised as an objection to any analysis of correlations among neurons, actually even more generally than in the hippocampus, because of course correlation structures change. Correlation structures in the retina change during adaptation. Indeed, what we're talking about is the behavior of some population of neurons under some reasonably statistically stationary set of conditions. The question of how you get from one stationary set of conditions to another is kind of a separate problem. I think it would be interesting to know, for instance, if you do an experiment on the same population of neurons in a very different environment, so that the system has adapted or remapped or whatever, in some sense, are you in the same, if we're allowed to use the language that sort of is in the background behind this, do you end up in something that's in the same universality class, or do you actually slide around? We've really I've told you you're kind of the first pass through, so I think these are good questions.
Great. Okay, we have another question now. That's from Mohsen Hadiampur. There's two questions. The first is, in your model, and I think this was referring to the second part of your talk, if I remember when the question was posted. In your model, do you consider neuron activities, or generally whatever your model response variable is, to be pairwise independent? If yes, could you elaborate more on the assumptions? Maybe we'll just start answering that question first, if it's clear enough.
So in the maybe this is one where a discussion would be helpful. So one of the reasons that, I think some of you will know that before we did this work on trying to do coarse graining in very large populations, we'd spent a lot of time trying to build explicit models for populations of order 100 neurons. And there, you have to make some definite assumptions about, what things you're going to take seriously and so on. And so, for example, one of the things you can do is to build models that match the pairwise correlations among neurons. You're not assuming anything about the higher order correlations. In fact, quite the opposite. By building the minimally structured models that match the pairwise correlations, you predict the higher order correlations, and they're non-trivial. And in the case of these kinds of experiments in the hippocampus, it was possible to show, and this was, I think, really a a beautiful part of Linoy's thesis that was published a couple of years before the article that I cited, that the higher order correlations are predicted essentially within the error bars with which they can be measured. And I'll remind you that when you have 100 neurons, there are almost 100,000 different triplet correlations to be measured. So the fact that you get them all right within error bars is really, I mean, we were quite surprised. So that said, an important part of what we're doing when we do the coarse graining analysis is we're not really assuming anything. We're asking what happens if you coarse grain in this way. And I should emphasize that this is one way of coarse graining. So the intuition is that things should get simpler as you coarse grain your description. And so we proposed one way of coarse graining, and we discovered that things simplified in the way that I showed you. It's by no means obvious that that is the uniquely correct way of coarse graining. And depending on people's interests, I mean, we can talk about this, or I could just put a period there. It's not obvious that that's the right way to do it. It's interesting that this simplification did occur. So you're not really so the fact that you use, for instance, pairwise correlations as a notion of neighborhood doesn't assume that higher order correlations aren't important. In fact, you'll notice that the probability distributions that emerge are strongly non-Gaussian, which means that there are non-trivial higher order correlations. We're just not using them in constructing our coarse graining procedure. So I hope that's clear.
Hopefully that answered. I'm not sure if we have the Mohsen with us, but there was a second part to this question, which is, how did you handle your model parameter estimation procedure? More specifically, did you use likelihood EM or something like Monte Carlo estimation?
So again, in the coarse graining process, there is no parameter estimation.
Right.
There is the worry that the correlations that you're using have error bars, and so you should be careful that your coarse graining procedure isn't just chasing the noise in your measurement of the correlations. So but that's a very technical thing. Maybe depending on exactly which part of the talk this was referring to, in building models for the sequences that we see in fly behavior, there the way you do it is you make a guess of this interaction function J, you do Monte Carlo, you check whether the predictions of your Monte Carlo simulation matched the pairwise correlations or not, and if they don't, then you move the J's in the direction that would give you a better match. And that that notion of giving a better match between the predicted and observed correlations is actually the same as doing maximum likelihood. So for the whole data set. So
great. Thank you. I'm going to skip down to a question by Siddhartha Mitra, because it also relates to correlations.
Mm-hmm.
And the question is, can correlation distances change over time dependent on change in behavior? Also invite on screen if.
Again, I think the short answer is yes. And I would say again, we've looked at periods which are statistically stationary. So the animal's engaged in one kind of behavior. I mean, it's not doing exactly the same thing over and over again, although maybe running along a track is a little bit too much exactly the same thing over and over again. That's a separate conversation. Yeah, so it's engaged in some ensemble of behaviors which is statistically stationary, and presumably if we moved to another ensemble of behaviors that was also statistically stationary, then the correlation structures that you see must be different in detail. And then the question is, well, which of those differences of detail would survive coarse graining? And we just don't know the answer yet. We've tried to do a little bit of this with populations of cells in the retina, but the experiments that my colleagues have done there go up to something like 150, 160, maybe 200 neurons, and that, I mean, really having 1,000+ neurons gives you a bigger dynamic range over which to ask these questions.
Okay, thank you. Okay, we're going to go back to, okay. Okay, there's another question here by Siddhartha. So I'll ask, if you got a network model consisting of neurons and interneurons, can we create a coarse-grained model consisting of these two groups separately, say just keeping the probability of them being connected the same?
So again, I think it's important to emphasize that for better or worse, we're not building a model.
Yeah.
So it's sort of like, I mean, I think it is useful to keep in mind the analogy between, suppose I showed you a molecular dynamics simulation of water, and somehow you know that there are these equations for fluid mechanics, right? So I show you what all the molecules are doing, and then you start, you say, well, I don't want to keep track of what every molecule is doing. I want to draw a box, and I count how many molecules are inside the box, and that's called the density of the fluid. And then I average their velocities, and I call that the velocity of that little part of the fluid. And then I do this on larger and larger scales. At no point have you actually written down a model for how molecules interact with each other. And for instance, there could be two kinds of molecules, and you decide to follow the analogy to two different populations of neurons. You could have two different kinds of molecules and keep track of the flow only of one of them, and just follow this process. And what we're asking is there something simple like the equations of fluid mechanics that comes out at the end? And we see signs that the answer to that is yes. And I would say, so the good news is we're not assuming anything about the nature of the interactions. I mean, there's a whole separate theoretical question: what must be true about the interactions in order for this to work? And so the good news is that we're not assuming anything. The bad news is that we're left with a kind of phenomenological description that says that you see these scaling behaviors, you see the emergence of these fixed distributions and so on, but we don't actually have a theory that tells you how that even describes all that. So I hope that helps as an answer to the question.
Okay, good. Maybe I'll ask just sort of a follow-up question, because I think there's maybe a little bit of theme here, but it's more of a very general question, which is relating to theoretical versus computational neuroscience, right? Because we're talking about building models, and I think there's sort of a certain mindset that approaches from a certain perspective of building models and data-driven and things like that, as opposed to sort of theory-driven, and not sort of looking at it from the same perspective. I mean, could you comment on that sort of generally? I don't know if that would be helpful. Do you have a philosophy?
Okay, so
about
let me literally triangulate. I mean, I can think about two very different approaches, right? So one is you really start with a mathematical description of the system at the microscopic level. So, ion channels, synaptic receptors, the whole apparatus, right? And then you ask, how do I get from that to, macroscopic behavior of the network that describes computational function, representation of information, and so on. Another approach is you could say, well, I'm uncomfortable about that because when I actually go try to do that, I've got lots of parameters that I have to set. And so I want some theoretical principles that allow me to cut through that complexity. And so here you have, ideas about optimization, you have ideas about learning and so on, that provide you with, in some sense, a response to a different response to the microscopic complexity. What we did here is a third thing, and I would say that it's the beginning of a path rather than, it's kind of dangling end, right? I mean, all these things are supposed to be connected to each other, right? That is to say, all that microscopic complexity really does accomplish some function, and it accomplishes it, and we have a metric for how well that function is accomplished, and so you must be able to tie those together. What we're doing here is asking, well, if I systematically try to zoom out and not describe the activity of every single neuron, but describe the activity of clusters of neurons that are brought together in some way that respects their functional behavior, namely their correlations, then are there hints that there's something simpler that might emerge? And I would argue that yes, there are hints. That's simply an empirical question. It doesn't have to be true. And then you could ask, well, maybe you should apply that same reasoning to the microscopic models. Maybe you should try and directly construct what that simpler theory is at the macroscopic level, but we're just not there yet. I hope that helps.
Yeah. Okay, I'm gonna go back to the questions here. We have one, I think this one's from this morning from Ahmed. Can you elaborate on the point you raised about how information can still be reliably measured even if the shuffled information indicates otherwise?
Okay, so I mean, I could show the figures, but I don't know that that's actually helping. So suppose that you have a signal whose correlations die away very, very slowly. And you observe it for a finite amount of time, long, but finite. And in particular, if the correlations really are scale invariant, then in some way, observing for twice as long, the correlations just get to be half as big. There's no characteristic scale beyond which everything becomes independent. So actually, to really do this, let's do the normal case. The normal case is there's some characteristic time over which things are correlated. If you record a signal for much longer than that characteristic time, then you have implicitly taken in a large number of approximately independent samples. Right? So they're correlated over some distance, but if you go many, many of those distances, then eventually they're independent of each other. And so if you shuffle, and grab two, or alternatively grab 2 points at random out of your data, then as long as they're not too close together, they're essentially independent of each other. And the number which are far apart is, of course, vastly larger than the number which are close together. And so you should get 0 for the mutual information, or some tiny thing whose structure you understand. The problem is that if correlations die very, very slowly, then if you pick 2 points at random, well, let's say if you've recorded for an hour, if you pick 2 points at random, on average, they're what? 20 minutes apart or something. So, but what if the correlations haven't decayed completely by 20 minutes? Well, then that means that if you pick points at random out of your hour, they're still a little bit correlated with each other. And so they actually have a non-zero mutual information. On the other hand, if you actually pick only the points which are 40 minutes apart, well, that's farther than the typical pair. And so their mutual information will actually be smaller than the shuffled one, right? Because the correlations will be weaker. And so one of the reasons I showed, so if you're puzzled by this, good, we were too. As you can imagine, this happened. You can imagine how this happened, which is that one day we were doing something not particularly with this data set, and we found we were very accurately measuring how mutual information as a function of distance was falling away. And then we said, oh, just as a sanity check, let's shuffle. And then we got something that was at this funny point. And then we said, okay, let's step back and study some completely understandable situation where we synthesize data that has power law correlations and everything else. So what I was showing you is actually from synthetic data. So there are no mysteries. So I hope that helps. It's this weird thing that we're so used to the idea that eventually we have independent samples. But if correlations decay very slowly as a power, then we never really do. As long as we're observing one long sequence, the correlations extend forever. And I mean, the classic example of this is what we're doing now, which is human language. So if you look at the correlations even between letters in written English, the correlations or the mutual information between two letters separated by, or two characters, two letters separated by n characters, dies very slowly as a function of n.
Right. So there is a follow-up comment by Abdullah. Isn't the solution here to shuffle across different replications?
Yes and no. So yes, in the sense that then you really will get 0, except that if the replications, so a common thing that is done, for example, in behavioral experiments on model organisms like flies and worms, is that the replication will actually be on a different but presumably genetically more or less identical organism. But as you know, there are individual differences even among genetically identical organisms. And so one of the other points I was trying to make was that when we go in the, and actually that problem exists even if you think about replicating with the same organism tomorrow. Well, it's tomorrow, and tomorrow is a little bit different than today for reasons that we don't completely have control over. I mean, obviously, for many things we do have control, but there's some things that we don't know the control over. And so when you think about animal behavior, when you have many replicates, whether it's because there are multiple organisms or because there are multiple instantiations of the same condition, separated by longer timescales, something happens in between. And so it's possible that the mean behaviors in these different things are a little bit different from each other. And as I showed you, even if 1% of the behavioral variance is ascribable to these individual differences, that distorts the measurement of correlation functions and things like that. So for the literal question, can you recover 0 for the mutual information? You're right. But the larger question, I mean, in some sense, the mutual information thing is just another way of sort of tickling your thinking about the challenges that arise because you never have truly independent samples in time.
Okay. We have another question here by Jung Young Kim. I will invite on screen just in case they do want to join us. But the question is, I understand that your study says that some universality exists in the brain. Is that right? What do you think the parameter that determines this universality, like critical exponent of correlation function, for example, in the Ising group, the critical exponent changes with dimension 2D versus 3D. Is it possible that neural network connectivity between ENI cells, its dynamic properties, or something else?
So this is a very ambitious question.
Yeah.
I think so the question points to things that we're thinking about, but are nowhere near being able to say. I've joked with colleagues that before universality, you'd like reproducibility. So so to keep with the example that all these different fluids that are made out of different molecules obey the same equations of fluid mechanics. I mean, we could do the precise critical exponent thing, but let's do the fluid mechanics one. The fact that it's the same equations of fluid mechanics for fluids which are different microscopically. So there is this kind of universality. That's wonderful. But the first thing you'd like to do is to show that the same equations of fluid mechanics describe this sample of water and this other sample of water and this other sample of water. And when it comes to samples of water, we feel reasonably confident that we'll get the same answer. When the new sample is a different mouse, everyone in this audience has dealt with the problem of reproducibility in these kinds of experiments. And so one of the reasons we're excited is that we don't just measure we don't just measure an exponent, which, by the way, it is tempting to think that these exponents we're measuring are like the critical exponents that we know about in statistical mechanics. And of course, I would love for that to be true, but I think you should be a little skeptical. I mean, we're not quite there yet. But the first step is, if anything like that reasoning is going to be right, it had better be true that we get the same exponent when we go into the same region of a different mouse's brain. And we do. As I say, in some cases to 2 decimal places, which is pretty astonishing. So that's the right direction, but we're not ready to say that we've found universality classes, because let's see the earlier questions about what happens if the animal were engaged in a different class of behaviors.
So I have a follow-up to that, and we'll see if the question asker has one. But I've seen you write elsewhere about irreducible sloppiness, and the fact that you can get these things, reproduce them to a decimal place or two, or what. Does that suggest that you're really on the right track in terms of what you're looking at, or does it give you more confidence, or
yeah,
is there still room for caution there?
So I guess this is like asking, should you be surprised by X, right? And at some point, whatever X is no longer surprising, right? For any X, right? So for me, the fact that you can reach that level of quantitative reproducibility was surprising. Similarly, as I mentioned, when we were trying to build models and found that you could reproduce the triplet correlations so precisely, that struck me as surprising. And I think your phrase that it makes you feel like you're on the right track, I think that's the correct interpretation. The question of whether any particular observation is the thing where you say, okay, now I'm sure I understand what's going on. I don't think that's a claim to be made by the person who made the observation, to be blunt, right? That's something for the community to decide, and I think it takes time. It takes time in my own thinking, right? I'm not sure whether how much of what being on the right track and being right are very different things. And as I think I've said already, the nature of our claims, we're not claiming that we have a theory of the dynamics of these networks. We're making a set of observations which point to the possibility of a simpler underlying theory. But we haven't held up the theory and said, okay, this is it. And then you could ask, are we right? I would say that the idea that this kind of simplification procedure has a chance of working in this rather more complex setting than where it started in statistical physics, I think, yes, we have evidence that we're on the right track for that, but it's a road. Right?
Okay, so the right direction.
Yeah.
Okay, good. Thank you. We have another question here from Daniel. I'll invite him on, but I'll read the question in the meantime. I see behind Bill the dual language Godot. If I scan a French, then the English page, would the same coarse-grained reduction apply? Except for action selection, do we know that the brain needs to do dimensionality reduction and a reticle suggestion?
So first of all, I'm impressed that you.
Oh, he's here. Okay, good.
I'm impressed that you can read the titles of the books on the shelf behind me. I'm not sure. Oh, I got
that and Spikes were the most visible ones.
I see. Well,
so your own book is there.
Yeah. So I think that there is evidence, for example, that although this could all be done better, there was a period in the 90s where people got very interested in these issues of correlation structure in written texts, and then it kind of petered out. And so I think actually there are things worth going back and doing, and I've talked to colleagues about this, and we've made some starts, but haven't quite gotten there. I think it is true that if you sort of measure the right correlation structures, then if you look at translated versions of the same text, you see the same kinds of correlation structures. Obviously, the roles played by different letters, it can't be that the actual sequences of letters are the same, because they don't parse in the different languages. But for instance, if you ask, how does the mutual information between the choice of letters or the choice of words decay as a function of separation, I think you find the same answer when you look at two bodies that are translated. If you mistakenly took the volume of Beckett behind me and didn't notice that you should do the analysis separately on the two different pages. Sorry, so for those of you who don't know, edition of Waiting for Godot, which is behind me, actually has facing pages in French and English. An extraordinary fact that Beckett did not write in his native language. Anyhow, so if you took time as running not through the English text and separately through the French text, but rather alternating from one to the other, you would see something very funny, obviously. And so the question is, are you in danger of making mistakes like that when you analyze the brain? And I think your point about action selection, of course, yes, you're in grave danger of making mistakes like that. And so that's why we study, for instance, some period in which the behavior is statistically stationary. And are we doing that right? No, hard to know for sure.
Well, let me amplify, because in the case of the dual language Godot, there are actually 3 different types of representations in the brain, and the coarse-grained reduction is probably different for the 3 of them. The first is the early vision one, where you see a white page with small black objects arranged in a line. The second level is the one you talked about, where you're talking about, say, letter frequencies and arrangement in words. But there's a third higher order level in which the sense of what Beckett is writing gets through. And whether you're reading it in French, that should be the same. So that was why I was trying to complicate that.
You're aiming for something that's, you're touching much harder questions.
But those are the fun ones.
Yeah, absolutely.
And that's not as provocative as my second question, because I sort of segued into the one. The question is, action selection obviously requires dimensionality reduction. You have to do something, and there's a limited set of choices in most circumstances. But I escaped from physics earlier than you did. And so the concept of reductionism, which I always favor, and dimensionality reduction, maybe the brain doesn't care about that at the levels at which we study neurons. And so all the sophisticated analysis we can apply might not apply to the brain.
In some sense, I would say that most of my talk was, with respect to the usual notions of dimensionality reduction, somewhat skeptical. So I'm not sure. I maybe I should just say yes and leave it at that. I think when in thinking about action selection and what that means about dimensionality reduction, it's a little bit like my straw man about, it only takes 100 muscles to talk. So does that mean that language is 100 dimensional? And as people commented in the chat, don't take this too seriously. But what's interesting is that when we say that about language, we have no problem immediately saying, no, of course not. You have a brain, right? And language is really a property of your brain. It's not, it is ultimately a motor act, but that's not the right way to think about language. But then, I don't know, when I think about a fly walking around, he's got a brain too, and he's certainly got a lot more neurons in his brain than he has motor neurons for his muscles in his thorax. And so why shouldn't there be a very large number of hidden internal states in the fly's brain, which indeed we see hints of when we actually try to analyze the behavior quantitatively, and where they show up is in these very long-range correlations again. So, as I say, short answer, yes. And longer answer, you've pointed to some places where one needs to be very careful in analyzing things, but I don't know that, I would say that the kind of analysis that we've been doing makes it more obvious that you need to be careful. I don't know that other analyses are exempt from those needs for care.
Great, thank you.
By the way, I don't think I've completely escaped from physics, or at least I hope my department chair doesn't think so.
Okay, I've invited Thomas up because he asked the next question. I don't know if I can, well, I'm going to read that question. The RG approach and universality work in the thermodynamic limit, is the brain large enough to be close enough to the thermodynamic limit? but I see that Thomas has joined us, so hopefully he can answer or clarify if there's necessary.
So even worse, when there's a question of whether the brain is in that limit, is the set of neurons that we record from large enough to be in that limit? And the answer is we don't know. And so we kind of flew by the seat of our pants. And I would say it is surprising that as we do this coarse graining, you kind of get on these scaling trajectories very early, right? So, you know, you go 1, 2, 4, right? And you think, okay, fine. By the time I get to 1,000, something interesting should happen. But actually, by the time you get to 4, you're doing pretty well. And I don't know whether that's good news or bad news. That is to say, it's good news in the sense that when I show you something scaling, I can show it to you over a very wide dynamic range. On the other hand, it might make you worry that something weird is going on that we're not understanding. And at the moment, we have access to populations of this size. And so that's what we can do. But great question.
So would you take your success in getting similar exponents as an indication that it's working? Yeah, so that you're actually in a scaling regime.
Well, the way I tend to say it is that what this means is that we've identified something about the collective behavior of the network that's meaningful because it's quantitatively reproducible from animal to animal. And then the question is, what's the interpretation? And that you know, I think you know what interpretation we'd like to have. And whenever I talk to a physics audience, everybody's headed in the same direction. Is that the right interpretation? I don't think we know yet.
Okay, yeah, thanks a lot.
Thank you.
Thank you. Okay, we have another question here from an unidentified asker. But it starts off not directly related to this morning's talk, but in one of your papers, you found that the retina efficiently encodes predictive information on visual stimuli. Do you expect this to be replicated in other sensory systems, for example, auditory, or has this been studied? Thanks.
So I find, you know, having spent a number of years of my life trying to understand, in some sense, you know, trying to approach the question of neural coding by asking, what is it, you know, when I see this response of neurons, what is it that just happened in the world that caused this response? I find myself now much more interested in the question of, well, if I'm the organism, I really don't care what just happened. What I'm interested in is what's going to happen, because that's where I get to choose, you know, when I choose my actions, when I decide what to do, that's always, my success or failure will be judged in relation to what's about to happen, not what happened in the past. And so, you know, this idea of trying to understand the way in which neurons encode predictions beyond, you know, very sharply defined issues of prediction of reward, I find very interesting. The paper you're referring to, which was led by Stephanie Palmer, was one sort of first foray in that direction. She and her colleagues. So actually, the group was Stephanie and Olivier Marr, who was a postdoc in Michael Berry's group at the time, and Stephanie and Olivier have continued to collaborate on this in the context of the retina. Olivier now has his own group in Paris, Stephanie in Chicago. But the question you really asked, the specific question you asked was, what about other modalities? And the answer is simply, I don't know. I don't know whether anybody has taken that up. It would be interesting. I mean, obviously, the thought is that there's something general about the problem of prediction. That's one of the reasons why I think it's intriguing. In particular, the problem of separating what the predictable part of signals from the unpredictable part of signals. That has a certain degree of universality, not in the technical sense of statistical mechanics, but in the qualitative sense. So yeah, I would think that some ideas like this would be more broadly relevant, but I don't have anything concrete to say about it.
Great, thank you. Are there any more questions? We have exhausted the question list, but we'll give a few moments if anyone in the audience has something they'd like to ask before we stop. But 40 minutes has flown by very quickly. Okay, I don't see anything popping up. So if there are no more questions, I would like to sincerely thank Bill Bialek for a great presentation. Oh, no, Daniel's back.
Oh,
Just
I see.
in time. Just in time. Okay, can you see the comment there, Bill?
Yes, yeah.
Okay.
I don't think it needs a reply. We were sort of joking about the notion of escaping from physics, but anyhow.
Okay, fair enough. Okay, so yes, thank you very much, Bill, for a wonderful talk and for taking all of this time to come back and answer people's questions. It's a beautiful day out, at least in my neck of the woods, and it is a holiday as well. So thank you for spending all of this time with us, and thank you to the audience as well for the great questions. We hope you enjoy the rest of the conference. The poster session, is starting soon, so please go and visit the posters and learn more and come back tomorrow as well. Okay.
Thank you. It was a pleasure.
Thank you very much.
Bye.
Bye-bye.